Hello and welcome everyone to this podcast for cell biology. In this podcast, we're going to introduce you to the cytoskeleton. We'll begin with a short definition of a cytoskeleton, and then we'll talk about the three kinds of cytoskeleton: actin, intermediate filaments, and then finally microtubules. But let's begin with a brief definition of cytoskeleton and some of the general functions of the cytoskeleton. Okay, we could define the cytoskeleton as this dense and dynamic complex network of protein fibers in the cell that function in the following. Some of the cytoskeleton helps maintain cell shape. Some of the cytoskeletal elements help in cell movement. And this is both intracellular, meaning moving things within the cell, and extracellular, that is, moving the cell from one place to the next. Third, the cytoskeleton helps in cell division. Fourth, it functions in cell contraction, mainly muscle contraction. And lastly, the cytoskeleton functions in the interactions between cells. So this is a brief description of the cytoskeleton in general terms and some of its functions. Not all of them, but some of them. Let's go ahead now and talk more specifically about actin. Okay, so let's talk about actin filaments. We sometimes call actin filaments microfilaments. And this is simply because actin filaments have the smallest diameter of all the three cytoskeletons. Let's list some functions specific to it. Allows a cell to crawl. We'll talk more in detail about this later on. But for now, you may have a cell like so, and it needs to move towards some kind of attractant, and it will protrude towards that attractant. Or maybe it's another cell it's trying to reach out towards. But the end result is that the cell moves. Next, the actin cytoskeleton helps engulf particles, or as we recently learned, it aids in the process of phagocytosis. So if there is a particle that the cell wants to engulf, it will take the cell and it will reach out towards it and wrap around it to bring that particle into the cell. Third, the actin cytoskeleton is important in cell division. As a cell completes its mitotic cell cycle and it divides in two, actin plays a role in pinching these two cells off to form the separation in these two new cells. The actin cytoskeleton also functions in the formation of microvilli. You see these in various parts of our bodies, but for instance, lining our gut, we have these protrusions sticking out to increase the surface area of these cells so it can absorb more nutrients. And they're made by having actin cytoskeletons push the membranes up, making these microvilli. Lastly, they're important in cell contraction, mainly that of the muscle contraction. Now that we understand the function of the actin filaments, let's now look at the structure and the formation of these filaments. So, actin structure and polymerization. When I say polymerization, I just mean the growth of the actin filament. The actin will form a filament like so. I'm just going to draw these circles here, and each of these circles represents one of the actin filament's subunits. Each of these individual subunits we just refer to as an actin subunit. As I said before, the actin filament is often referred to as the microfilament, and that's because its diameter is often the smallest, and it's usually about 6 nanometers. Now the actin filament, as I've drawn here, has a polarity to it. At this end, we call it the plus end, and this end, we call it the minus end. At the plus end, these subunits of actin contain ATP. So I'm going to draw this as actin ATP. The subunits going towards the minus end have actin ADP. Individual subunits, as they bind to this plus end, they will have ATP attached to them. And eventually that ATP becomes hydrolyzed as the actin attracts more and more subunits. Actin subunits could add both to the positive or the minus ends, but there is a strong preference to binding at the positive end of the actin filament. One thing I forgot to mention was something about the actin subunits. And I mentioned in your note-taking guide, so I want to make sure I state it here, and that it's overall abundance. Actin, these actin subunits, are about 5% of all proteins in an animal cell. They are about 20% of proteins in a muscle cell. Now, with the idea that there's so much actin in a cell, one might think that these actin subunits could continuously bind to this positive end and keep growing, but that's not what happens. So I'm going to draw over here a cell, and in this cell, I'm going to draw various actin filaments growing in different directions. They each have their positive ends, and they each have their negative ends. Within the cytosol of this animal cell, you will see many subunits. So much that at this critical concentration, one would predict that these subunits would automatically add to these positive ends, but they don't. There are two strategies to prevent growth, or I might say to regulate growth, because it's ideal to have these subunits in the cytosol at all times so that when you want to grow the filaments, it's easy to grow them without having to make the proteins. But you don't want them always to add. So what do these cells do? As I said, there are two strategies. The first is to use this protein called profilin. Profilin's not the only protein that can do this. There are several others. But for simplicity, I'm just going to talk about profilin. Now what profilin does is it will bind to each of these subunits, and as long as profilin is bound to these subunits, it is unable to add to the growing microfilament. So I'm going to draw it right here, profilin, bound to actin subunit, can't be added. The actin filament. The other strategy is that various proteins that we're going to call capping proteins bind to the plus end of the microfilaments. And when they're bound to the plus ends of the microfilaments, they block polymerization. Now when the cell does want to have actin growth, it will remove the profilin and the capping proteins so that actin subunits can be added to the positive ends. This is just a strategy to have an abundance of actin subunits in the cytosol waiting to be added. I want to talk now about one of the functions of the actin cytoskeleton, and that is cellular crawling. Alright, so let's talk about cell crawling. So why would a cell want to crawl? It's a good question. A cell wants to crawl, that is move from one spot to the next spot, for various reasons. In eukaryotes that have a brain, it's important in neuron development. Generating those very long neurons we have in our bodies requires cell crawling. It's also important in amoebas, as an amoeba will begin to move in one direction to get it to its desired location. They're also important in neutrophils, which are part of our immune system. And neutrophils are important in identifying, engulfing, and degrading various pathogens that enter our system. So I want to demonstrate this by talking about the formation of something called a lamellipodia. This is something the amoeba would do. And so we have our cell here, and the actin cytoskeleton will begin to form filaments moving in one direction. As they push towards this plasma membrane here, the actin cytoskeletal filaments here will push that membrane further and further away. And this is what we call a lamellipodia. What happens at this point here is the lamellipodia will anchor to the surface And once it anchors to the surface, the rest of the cell will release its end here and slowly move in this direction. Then next I want to talk about something called filopodia. And this is when you have your cell here and the actin cytoskeletal filaments in a more concentrated area send out a narrower strip of the cell. This is really important in neuron development. I didn't draw it, but this will have the actin cytoskeleton moving down this cell, protruding it in this one direction. I'm going to show a quick video here of what this looks like, and then we'll come back and finish our discussion of the actin. What we see on the right half of the screen here is a human neutrophil. And over here we're going to see a pipette that's going to place some chemicals here that it's going to attract the neutrophil. And as it's released the chemicals, you'll see the neutrophil moving towards these chemicals. What we see are those three steps. We saw the protrusion of the cytoplasm moving forward, and then we see the whole rest of the body of the cell moving with it. Again, moves forward and the whole body of the cell moves with it. Pay attention to how it protrudes the cell membrane and how the rest of the body of the cell moves forward. And in this particular example, they're being a little cruel with these neutrophils and making them move in opposite directions. But you can see how quickly they can change directions based upon the chemical signaling. Okay, welcome back. Now, to finish our discussion of actin, I want to talk about actin motor proteins. And as the name implies, these help things move along the actin filament. Remember earlier on we said that various things within the cell, like organelles, they can move along the actin cytoskeleton. But they require specific motor proteins to move things. These motor proteins are called myosins. There are about 20 different myosins that have different tissue specificities and have different functions. But remember, as they move, they will always move towards the positive end. So what do these motors look like? Real briefly, they will all have this end here that we call the head domain, and then they will have a tail, which cleverly enough is called the tail domain. The head domain binds actin and ATP, and it will also hydrolyze ATP. And ATP, as you might guess, is the energy source for motor movement. This tail domain has lots of variability. It is what helps distinguish the 20 different myosin motors. And the reason it has variability is because the tail domain binds various cargo. It may bind a mitochondrion, it may bind some other organelle, it may bind a different protein altogether, but this is what gives this motor specificity. To show how these motors work, I want to show a quick video, and then we'll come back. So as I start this video, in green here are the myosins, in red are the actin. Attached to the actin is the head portion of the myosin, and the tail portion here is attached to what is here is just a glass slide. But this could attach to various kinds of cargo or to various structures in the cell. As we see here, ATP is added onto this cover slip, and ATP will bind myosin, and as it's hydrolyzed, it will change its conformation set in such a way that it moves the actin forward. And this is showing the actual data. And you can see the actin, I'm going to pause it here, you can see the actin kind of moving around this slide, almost like a snake cruising around here. And what's not shown here are the myosin motors that are attached to this slide, moving it in this direction. Okay, well that ends our discussion for the actin microfilaments. So let's move on to intermediate filaments. So as we begin, let's talk about some characteristics of intermediate filaments. Their main function in the cell is to protect against mechanical stress. And we'll talk more about that in a little bit. They also play a pretty important role in chromosome stability. And we'll talk about that in a moment as well. Now, the reason they're called intermediate filaments is because when they were first identified, they had a diameter in between the sizes of actin and microtubules. They're also extremely stable, these intermediate filaments, which is probably why they play a role in protecting the cell against mechanical stress. But these filaments are the strongest of the cytoskeleton elements. They're well distributed throughout the cell. We're going to talk in a little bit about their role in the cytoplasm as they help hold cells together and in the nucleus. But before we do that, I want to talk about the structure of intermediate filaments. The subunit of intermediate filaments we can simply call intermediate, and I'm going to abbreviate it as IF, intermediate filament proteins. We'll talk more specifically about the kinds of intermediate filament proteins, but for now we'll just call them intermediate filament proteins. They're fairly long proteins compared to some of the other cytoskeleton proteins or subunits, and they have an N-terminus and a C-terminus, as you might predict since it is a protein. And so what we're going to do is we're going to take two separate intermediate filament proteins, like so, and what they will do is they will form a dimer. And in doing so, they will wrap around each other in sort of a helical pattern. And this we call the intermediate filament dimer, because there are two of these proteins. So we call them a dimer. Now, this isn't what gives it its strength, having one of these. What gives it the strength is when these dimers form a tetramer. Two separate dimers come together, and they don't bind right on top of each other, but rather they're staggered. And so let me try to demonstrate that here. We still have our N-terminus and C-terminus, as always. And then these tetramers here, like why don't we write that here, these two dimers together equals the tetramer. They will then bind to other tetramers. Also not right on top of each other, but in some kind of staggered pattern. And they will eventually form a tubule of sorts that wraps around each other. And in between these, we have certain non-covalent interactions holding them together. Now, this much larger structure of intermediate filaments provides the cell with the strength it needs to withstand various kinds of stresses it might encounter. Let's talk about some examples of intermediate filament proteins. And we can think of these as subunits as well. I'm going to list 4 of these here. The first is keratin. The second is something called vimentin. The third are neurofilaments. And the last are nuclear lamins. The first 3 here are all found in the cytoplasm. I'll put a C by them. This last one is found in the nucleus. And in just a moment, we'll talk a little bit more about keratin and a little bit more about nuclear laminins. But before we do that, I want to give some examples of where we would find these different kinds of intermediate filaments. Keratin we find lining the gastrointestinal tract. It's found on the tongue, in the tongue, cornea of the eye, hair, and feathers for birds. Vimentin we find in muscles and glial cells. Glial cells are found in the nervous system, but they are not neurons. They are supporting cells in the nervous system. Neurofilaments, these are found in neurons. And the nuclear lamins, as you might suspect, are found lining the nuclear envelope. So I want to talk just a little bit more about keratins. Now, what keratins do is they provide that protection against mechanical stress. And briefly, how they do that is that they help stabilize neighboring cells. So let me just draw 3 cells next to each other, and we'll have our keratin fibers here extending between the cells, or I should say within the cells, because they don't actually protrude out of the cell and connect to the other cell. But what they do is they interact with a protein complex at the membranes. So the keratins bind to this protein complex that does extend across the plasma membrane, and it interacts with the intermediate filament of the neighboring cell, and so on. I won't ask this on a test, but just in case you're curious, these are called desmosomes. But desmosomes are the proteins that hold these cells together and help in the coordination of the keratins from one cell to the next cell. All right, next I'd like to talk about the nuclear lamins. And for this particular drawing, let's pretend the whole whiteboard is the cell, and I'm going to draw the nucleus like so. So this is the nucleus. The nuclear lamin proteins will form a network of intermediate filament proteins, that is the nuclear lamin proteins, along the inside of the nuclear envelope. These nuclear lamin proteins are often referred to as the nuclear lamina. Now what do? They help provide some structure of the nucleus, but we're learning more and more about a very important role they have, and that is that they bind to chromosomes. So the chromosomes are attached to the nuclear lamina. Now these have to release the chromosomes or parts of the chromosomes at various times, and those times include during DNA replication and transcription, the process of making RNA from the DNA, which we won't talk about in cell biology, but you will in genetics. So during these times, it will release part or all of the DNA from the nuclear lamina so it can be replicated or transcribed. Okay, let's go ahead and move on to our third and our last part of the cytoskeleton, and these include the microtubules. I'm going to briefly talk about three different functions of the microtubules, thinking about three different kinds of cells. In a non-dividing cell, microtubules play an important role in moving organelles and other cellular structures along these microtubules. Microtubules always originate in their development through something called the Microtubule Organizing Center, or just MTOC. And from the MTOC, microtubules will grow. And they may attach to various organelles, not always the same organelle, but different organelles, and help move the organelles in various directions. Next, I want to talk about what microtubules are doing in a dividing cell. We haven't talked about mitosis yet, but you may recall from a high school biology class how chromosomes are lined up along the middle of the cell. Now you may also remember that there were these two structures on either side of the cell called centrosomes. And inside each centrosome were a pair of structures called the centriole. These centrioles are bundles of microtubules. And the role here is they serve as an organizing center to send out growing microtubules to attach to the chromosomes. These microtubules, once they attach to the chromosomes, will begin to retract and pull these chromosomes towards the other end of the cell, and ultimately what will happen is the cell will divide and you'll have two new cells with equal complements of the chromosomes. But we'll talk more about that when we talk about mitosis. The last one are the ciliated cells, where you'll have a cell like so with cilia protruding out. And these cilia are formed because of microtubules that push out, forming the cilia and also allowing the cilia to function in a way to have their movements move back and forth in a swishing motion to help, in some cases, bring particles closer to the cell. I should also mention, though I didn't draw them, the microtubules would also be important in the formation and the function of flagella. Okay, next let's talk about microtubule structure. First, let's talk about microtubule subunits. The microtubule subunits exist as a dimer, rather a heterodimer, because you have two different subunits that come together. You have the beta subunit and you have the alpha subunit. So how do you begin to form the microtubule? Well, you'll take these subunits and you will bind them to each other like this, where you have one heterodimer here binding to another, to another, to another, until you have a long filament. Now the microtubule, unlike the actin, isn't just a single filament, but rather it will form additional filaments next to it. In fact, it will form 13 of these filaments that we call microfilaments. So 13 microfilaments. And these microfilaments, as they're being added, will wrap around each other, forming a tubule. So these 13 microfilaments form one microtubule. This one microtubule is about 25 nanometers in diameter. So of the three, it is the largest. I did forget to mention, just like the actin filament, the microtubule has a plus and a minus end. And these heterodimers, when they come in, they like to come in at the plus end. Just like with actin, it can come and bind to either end. There's just a higher preference to binding to the positive end. And what stabilizes these ends, it's not ATP that's bound to them, but rather it is GTP. And as you move down, you will have more of the subunits bound to GDP. So for higher stability, GTP is associated at the plus end, and this is where the new heterodimers come in as the microtubule grows. Since microtubules have an important role in cell division and the movement of structures inside the cell, it's often a target of pharmaceutical companies. And two drugs that affect the stability of the microtubule are Taxol and colchicine. Taxol stabilizes microtubules, whereas colchicine destabilizes. Now, first thought, you might think stabilizing the microtubule isn't such a bad thing, but it's important to remember that for the microtubule to function properly and for the cell to survive, the microtubule needs the ability to grow and to shrink. And so Taxol prevents the shrinking part of the microtubule, whereas colchicine prevents the growing part of the microtubule. Okay, now I'd like to talk about microtubule motor proteins. Just like actin had myosin as a motor protein, microtubules also have motor proteins. They have two classes of motor proteins, microtubules do. The first motor is one called kinesin, and the second one is called dynein. In a general sense, I want you to know that kinesins moves cargo towards the positive end. I always remember this because kinesin, I think of kinetic energy, and I think of kinetic energy as a positive thing. So kinesin moves it towards the positive end. Dynein does the opposite. It moves cargo towards the negative end. And I associate the word di with a negative. So that helps remember dynein moves it towards, is the motor protein that moves it towards the negative end. So if you were to have a microtubule, like so, with a positive end over here and a negative end over here, you could have a kinesin, like this, that binds to the microtubule and then has a cargo on top here. And it will move this cargo towards the positive end. Likewise, you could have a dynein, like so, binding a separate cargo, but moving it towards the negative end. And we will see on any given microtubule both kinesins and dyneins moving cargo in opposite directions.